Okay. First of all, thanks a lot for inviting me for this presentation. So sorry, it's about bacteria, but the objective of the study I will present was to predict, to see to what extent we can predict the response of soil bacteria to multi-factorial global change. And for this, we wanted to explore the ability of trade-based approaches to do this. Okay. So as you know, we have in the literature many studies looking at changes in the activity, diversity, abundance of bacteria or fungi to global change factors. Very often they are looking at a single factor. So a first question of interest is, is to look at the effect of several global change factors at the same time and their possible interactions. That is when you have elevated CO2, elevated precipitation and nitrogen deposition. When you have all these together, which is the case in the Anthropocene, will you have only additive effects, very simple additive effects? Or do you have more complex and more difficult to predict interactive effects? And a second question is to try to go beyond observation of the responses and try to go towards an understanding and prediction maybe of these responses. And for this, of course, you can do this only if you are able to identify the key environmental drivers of the responses, the biological mechanism which underpin the changes observed. Okay, so for this today, I will focus on one group of bacteria in the soil, which are nitrifiers, and actually the functional group I will talk about, it's NOB for NO2 oxidizing bacteria, and they catalyze the second step of nitrification, that is oxidation of NO2 into NO3. So you can see this as a model group for this presentation. So in the soil, we know that there are two main genera of NOB, which are Nitrobacter and Nitrospira. And the specific objective of the present study was first to measure, to quantify the effect of three different global change factors, which are elevation of atmospheric CO2 concentration, changed in precipitation regime, and increased nitrogen deposition. These are three among three from the main drivers of global change today. So we wanted to look at their effects on NOB at the activity level, but also abundance and diversity, and to find a way to look at their effects as main factor, but also to see to what extent we can have interactive effects between these three factors. And the second question was to what extent accounting for the main functional traits of those guys can help understanding and predicting the response to global change scenarios. So for the first question, what we have used is we have used a great experimental design set up by Californian colleagues. So they are from the Carnegie Institute of Stanford, and what they have done, they have set up a long-term experiment in Mediterranean grassland, manipulating three factors, that is CO2 atmospheric concentration, precipitation, and nitrogen deposition. And for each factor, you have two levels, that is ambient and modified level, and the level is chosen according to global change scenarios. And so it's you have the values here for CO2, it's plus 300, precipitation, it's not only an increase of precipitation, but also modification of the rainfall period according to the forecast, and for nitrogen deposition, increased in nitrogen deposition. And because we have 3 factors and 2 levels per factor, this means that we have used 8 treatments with 6 blocks, that is 6 true replicates for a total of 48 plots. The measurements we have done, it's so the potential activity of NOB in the soil, their abundance, distinguishing the abundance of Nitrobacter and Nitrospira. We have then measured the dominant populations of Nitrobacter, not really their diversity, but the dominant populations. And then we have a range of measurements to characterize the environmental conditions in soil, in particular as regard to water content, carbon availability, nitrogen availability, including measurements of gross ammonification using a 15N dilution technique. So in terms of results, first, looking at the potential activity of this group, NOB, you have here figures with the value of the activity under each of the treatment here. First thing you can see, comparing the 2 figures above, between 2 different years, you have very similar patterns. That is, we have very clear treatment effects, and these treatment effects are relatively consistent across time. And the second message you can grab here is clear treatment effects like elevated precipitation decrease the activity of NOB. You have a tendency to have a positive effect of CO2, and very interestingly, you see here that there is indeed interactive effect between the 3 factors. This means that you cannot predict from single factor study, you cannot predict the effect of the interaction. And then in terms of abundance of NOB, and here it's the abundance of, on the left, Nitrobacter, on the right, Nitrospira. Again, comparing the 2 figures on the left and the right, you clearly see that the 2 groups, they respond very differently to the global change treatments. So they don't have the same ecology and they respond very differently. This is highlighted here in the statistical results with a tendency of negative effect of the elevated precipitation treatment for Nitrobacter, whereas you have a positive effect of nitrogen. And here for Nitrospira, negative effect of CO2, marginal, and a tendency to have a positive effect, not a negative one, of precipitation. So very different responses. And then digging into the composition of the Nitrobacter community, you have here a phylogenetic tree of the sequences retrieved from the soil under different treatments. And what we observed is that for some populations corresponding, for instance, to this major cluster here, you can see that these sequences, they are present in all the treatments. So these correspond to populations that are present, whatever the type of global change treatments we applied. On the reverse, we had 2 clusters, but in particular this one above, with sequences corresponding to populations that were only present under, let's say, 2 treatments, mainly the elevated CO2, elevated nitrogen, elevated precipitation treatment, and to a lesser extent, elevated precipitation treatment. So you see again a very clear treatment effect on the composition of Nitrobacter with a very clear interactive effect between the 3 factors observed. And very interestingly, the populations corresponding to the cluster above, they were affiliated to Nitrobacter hamburgensis. And you will see why it's interesting, because for those who know NOB, Nitrobacter hamburgensis is known to have specific functional traits, and in particular mixotrophic capacity. So just to tell you that it's not figures like this, it's significant. You have a very, very clear effect of this treatment. And as you can see, the population corresponding to the cluster A, affiliated to Nitrobacter hamburgensis, they were, let's say, they were not representing 1% of the community in most treatment, whereas in the last treatment here, you see they represent 63% of the community. So a huge change in the composition of the community. And it's very interesting because it's consistent with what I showed you before in terms of effects on the activity. I've stressed the interactive effect between the three factors on the activity of NOB, and you see that we have here an interactive effect very clear between the three factors on the composition of Nitrobacter. So the conclusions we can raise from the field observations, so interactive effect of global change factors on the, at least, activity and diversity of NOB, Nitrobacter and Nitrospira having different responses to the global change treatments, and the diversity within the Nitrobacter genus is clearly affected by the treatment effect, with this huge increase in population affiliated to Nitrobacter hamburgensis under the complex CNW treatment. So the question, second question in this study was, can we go beyond observation? Can we try to understand? Can we try even maybe to predict the observations of the global change factor effect we have here? And to what extent we can use a functional trait-based approach to help us to improve our understanding and prediction of the treatment effects. So I don't know if all of you are, well, you know quite a bit on trait-based approaches. For those who are interested, we have made a synthesis of trait-based approaches and their usefulness and limits also for better understanding microbial biodiversity and ecosystem functioning relationships. You can have a look. For the present study, the way I can present it is that, in my opinion, trait-based approach can complement very well other approaches we used, in particular molecular approaches, which can help us to characterize the modification of the soil bacterial communities in terms of composition, abundance, and so on. And then you can use trait-based approach to try to gain a better understanding and prediction. And I see this as a three-step approach. First step, you need to classify, well, to find a way to classify the different bacteria you have in soil thanks to their functional traits. And then you need to use this to inform a trait-based model that would be able to reconstruct, to represent the niches of the different functional types of bacteria. And if you have this, then you can apply your model to try to predict the responses. So in the case of NOB in soil, we have considered three main groups. First aspect is to differentiate between air strategist and car strategist, or oligo, or you want to call it. And this corresponds within NOB mainly to the dichotomy between Nitrospira and Nitrobacter. Nitrobacter are known to perform very well under high nitrogen, whereas Nitrospira, they perform poorly under high nitrogen. And the second dichotomy here within the first group is to distinguish between bacteria having a mixotrophic capacity and those who don't have mixotrophic capacity. When you do this, you obtain three groups. A way to present it is we split NOB between Nitrospira and Nitrobacter, not because their name is different, because their functional traits are different. And then Nitrobacter, we split them between these two groups. So, the first step was to categorize the different groups of bacteria. Well, you can use also a continuum if you want. Here it's a discrete approach. And the second step is to inform a modeling approach in terms of traits. So I've been working with Nick Buskill on this, using his modeling framework, and we have adapted it for NOB. Very shortly, this model is a mechanistic model. I do not have time to detail, but it represents energy acquisition and carbon acquisition, and finally biomass development. So the model considers the three groups and their competition and the output of the competition. So using a literature survey, we have informed the trait values for the different NOB functional types. And then, using this, you can test the ability of the model to simulate, to reconstruct the niche of each group of NOB. And very briefly, from the top to the bottom, you have from high to low level of oxygen, and in each subpanel, you have a range of carbon availability and a range of NO2 availability. So just two messages from there. For chemolithotrophic Nitrobacter, the model clearly simulates the major importance of N availability. This is what is important for them, and they perform well under high nitrogen. For mixotrophic Nitrobacter, they perform best under relatively low oxygen concentration, relatively high carbon availability, and high nitrogen availability, which is what we have in the literature. And the last part, Nitrospira, they are there, and they are not so sensitive to the range of conditions we have. So this is very interesting because this is very consistent with what we have in the literature. And the last part, the last step of the modeling approach is to apply the model then, prescribing the observed environmental conditions in soil, and try to see to what extent the result of the modeling approach is consistent with the observations, to what extent the model is able to predict the observed responses. So don't have time to detail, but you see that for the three groups, that is chemolithotrophic Nitrobacter above, mixotrophic Nitrobacter and Nitrospira, the model simulates, as observed, always abundant, high abundance of Nitrospira, but changes in the relative composition, relative abundance between Nitrospira and Nitrobacter. And here you have the CNW treatment with an increase in the mixotrophic Nitrobacter, and the model is able to grab this. And my last slide is to say that we have applied the model also to test the effect of global change on the activity of the community. And so you see again that the model is able to predict relatively well the changes in the activity. Very interestingly, when you disregard functional diversity within Nitrobacter, that is, you represent only Nitrospira and Nitrobacter, the model is not able anymore to properly simulate the activity change, and in particular, under some treatments, the model predicts very low activity, whereas we have activity in the field. So this shows the importance of expliciting the functional diversity. So my conclusions, it's, global change treatment, very clear global change treatment effects on those guys, with interactive effects between the different factors observed. The NOB community is always dominated by Nitrospira, but the percentage of Nitrobacter changed according to treatment. And you have putatively mixotrophic nitrobacter, which are strongly promoted under the CNW conditions. And the trait-based approach, modeling approach, allows pretty good prediction of the observed responses. You have seen this, in particular with this very interesting response here. And maybe the main message is representing functional diversity within the nitrobacter was needed to properly simulate the response of NOB to global challenge treatment and their activity. Clear demonstration of the importance of functional diversity for soil microbe. So thanks a lot for your attention. Thanks to my collaborators, which were not on the main slide, and I use the opportunity to advert for 2 postdoc positions in our team. Thanks a lot.
Thank you very much, Xavier. Maybe we have time for a quick question. Anybody? Nobody? Oh yes, on that corner.
So a bit of a... Is this... Okay. So a bit of a technical question. The assay that you do is a potential one to measure nitrite oxidation. And Nitrospira and Nitrobacter have different nitrite sensitivities. Do you think that you're biasing their contribution to the function by using a potential assay?
I would say that we have maybe the response thanks to the modeling approach. It is the result of the modeling approach is that even Nitrospira, there are relatively numerous. The key part of the activity, that is NO2 oxidation in soil, the main part is due to Nitrobacter. So that's a point, and I think it explains why we observed. It's not the first time we observed that the changes in activity, they are not linked to changes in Nitrospira abundance. They are always linked to changes in Nitrobacter abundance in soil, not in all environments, but in this type of soil. So that explains why. Yeah.